Hello, everyone, and welcome to the pre-lab lecture on the medicinal chemistry experiment entitled The Synthesis of Penicillin Analogs and Its Biological Activity. Okay, so I thought that the discovery of penicillin fits the Neil Armstrong's quote, one small step for man and one giant leap for mankind. Certainly the discovery of penicillin changed medicine in a very profound way and saved a lot of lives. So I think it's a very significant discovery. We're going to watch a video about the discovery of penicillin. Okay, so we are going to be doing an experiment where we are going to be making derivatives of penicillin, or changing part of the structure to see if the molecule retains activity or loses activity as an antibiotic. This is the core structure of beta-lactam antibiotics. Penicillins are not the only type of beta-lactam antibiotic. They're just one of several different structures that generally contain a fused bicyclic ring. In this case, we have a 4,5 fused bicyclic ring. And what makes it a beta-lactam antibiotic is the beta-lactam ring, which is this ring here. So you guys have heard about amides before, right? An amide, or sometimes people call it an amide. That is the functional group that contains a nitrogen next to a carbonyl. So if we have a cyclic amide, we actually call that a lactam. That's where that name comes from. And it's this four-membered ring that is, as you know, four-membered rings are unstable. They have a lot of ring strain, and so it's very reactive. And we're going to find out what it does, right? So they generally work by inhibiting cell wall biosynthesis in the bacterial organism. And these are definitely one of the most widely used groups of antibiotics. We're going to watch another video that shows the mechanism of action of beta-lactam antibiotics, and also talks about resistance to those antibiotics that is developing in bacterium over time. Okay, so you guys just learned about the mechanism of action for beta-lactams and how they inhibit cell wall synthesis, leading to lysis of the bacterial cells. And then also about the importance of resistance that is developed to certain antibiotics. So if there's resistance to antibiotics that we're using, for example, amoxicillin, then we need to come up with other antibiotics that the bacteria are not resistant to. Otherwise, we won't have antibiotics to use, which would certainly hurt our ability to treat different infections. So that's kind of brings us into the point of today's experiment. We're going to be looking at, well, if one antibiotic isn't working, we want to have some in the pipelines coming up that might be able to replace it. So how do we do that? So we essentially, what we want to do is come up with other structures that would work as well as the original. And so this is something that medicinal chemists would do. They make analogs of a certain compound by varying different chemical parts of a structure. So what we're going to be varying is the R group. We're taking the starting material, which is called 6-APA, which stands for 6-amino-pentylenolic acid. And we're going to be acylating the amine group with an acid chloride. And we're going to use different acid chlorides so that every group in the lab is making a different analog. And you can see all the different potential analogs that we're going to be making in the lab. So every group will be assigned a different one to make. They'll all be tested for their biological activity and their ability to inhibit bacterial growth. And we're going to use both a gram-positive and a gram-negative bacteria to see if it's effective against one or the other or both. And then the idea is to hopefully eventually do what's called a structure-activity relationship. And we'll get into that in just a little bit. So after we make the second amide, then we want to convert this into something more water soluble. Because if we want the molecule to be able to survive in an aqueous environment, then it shouldn't be nonpolar. We want to make it more polar. And so that's where we're going to take advantage of the fact that there's this carboxylic acid functional group that we can ionize. We can deprotonate it, make the potassium salt, which will be much more water soluble, and it'll be ideal for biological testing. Okay, so we're actually not going to be doing the biological testing ourselves. We're collaborating with a biotechnology class here at Miramar College. So this collaboration involves the organic chemistry lab, which is you guys making the compounds, and then they do the biological testing for us. And then both that class and our class can look at the plates after and can see the results of the biological testing. So on our plate, we're going to, actually the biotechnology class will divide it into 4 quadrants. They're going to put filter paper that's designed to be able to absorb some liquid that's put on it using a micropipette. So 4 pieces of filter paper that can absorb 50 microliters of liquid. And then we're going to test a negative control, which is just water, a positive control, which is amoxicillin. This will actually be used at 10 times less concentration than our compound. If we tested it at the same concentration as the compound, it would overwhelm the plate. The zone of inhibition would just be too large. So while our compound will be tested at 4 milligrams per milliliter, the amoxicillin is given at 0.4 milligrams per milliliter. That's just something to note when we're comparing our zones of inhibition, which is a measurement we can take measuring the diameter of the observed zone of inhibition. And we're also going to test the starting material, the 6-APA. So before we acylated, right, is this compound active against bacterial growth or is it not active? So was that acylation necessary for activity or is it not necessary for activity? We're going to also find that out as well. So when we get our plates back, hopefully they'll be labeled properly. So we'll know whose class the plates came from since there's multiple sections of 233L. And then also have some initials on there so that you know whose compound were tested on this particular plate. Okay, so our goal is to compile class data because every group is going to make one analog, but we want to know what all of the analogs activity is so that we can determine what's called a structure-activity relationship. So one thing you need to understand is that the structure determines the activity, right? So if the structure, if we want to know which parts of the structure are critical for activity, we need to do this type of structure-activity relationship testing, where we vary different parts of the chemical structure and test those compounds to see if they're see what their biological activity is. So for penicillin analogs, there's been lots of structure-activity relationship studies that have been done. such that they know that the stereochemistry at these two carbons, both hydrogens being back, is critical. The sulfur is actually not critical towards the activity, so it can be replaced with like oxygen or even a methylene. But this fused bicyclic ring and the four-membered beta-lactam is critical for activity, including this carbonyl and the carboxylic acid. These are important for binding to the protein. These methyl groups are important for biological activity. So what we're going to be studying, however, is what effect does varying the R group have on the activity. So if you look at the structures from the previous slide, you can find some structural similarities. Notice how in D, the carbonyl is directly bonded to an aromatic ring, whereas with, for example, I'm going to move my little thing so I can see all of them here. For example, with B, the carbonyl has two carbons before the aromatic ring, and those are structurally rigid because of the pi bond, as opposed to not structurally rigid and having more free axis of rotation. A here would have a carbonyl and then a nitrogen, and then directly, or sorry, I believe it's a methylene, can't see on my screen here, and then directly attached to a different aromatic ring. So you can see, we want to see what sort of spacing between the carbonyl and the aromatic ring is important for activity, right? Do we even need an aromatic ring? If we test I, would this compound also be active? What about halogens, right? If we introduce halogens instead of an aromatic ring, 1 or 2 chlorines, would those compounds still be active? So those are the sort of questions we're hoping to answer by doing this structure-activity relationship. So I wanted to give you guys an example of what can be found out by looking at structure-activity relationships. So we're looking at paclitaxel, which is branded Taxol. So you can see it's a very complex structure here. It's actually naturally derived, so they found it in the bark of yew trees. And it has certain parts that are significant for activity. So for example, they noted that this benzoyl group is required for bioactivity. So by replacing it with other groups that are not structurally similar, they noticed a decrease in activity. What about this hydroxyl group or alcohol group, right? It is also contributing significantly to bioactivity. So is this hydroxyl group. However, this phenyl group can be replaced with an alkyl group or even an alkenyl group, and you can get even better activity. What about this phenyl group? Well, they noted that after making several derivatives, that it's still active if it's replaced with a tert-butoxy group, which would be an oxygen and then a tert-butyl group, right? But the side chain here is important for activity. So this, you can't just take this entire part off and still have an active molecule. This acetoxy group is important for conformation. So it helps define the conformation, right, which is sort of the 3D structure of the molecule. And conformation is important for binding to enzymes, right? And then they also looked at this 4-membered oxetane ring, and the presence of it increases activity. So they have all of this information about what can and can't be changed about the structure to create different analogs that are still active versus rendering it inactive.